or the main office. They make decisions, things happen. All right. Endoplasmic reticulum, figure 7-8. The endoplasmic reticulum synthesizes protein for export from the cell. Oh, wow. That's good. That's what the endoplasmic reticulum does. Kind of looks like this, kind of like flat pancakes. You've got ribosomes that are stuck to the endoplasmic reticulum. And, wow, it's magnified 40,000 times here. Okay. Ribosomes. One of the most important jobs in the cell. Carried out in cellular factory is making proteins. Proteins are assembled on ribosomes. Ribosomes are small particles of RNA and protein found throughout the cytoplasm. They produce proteins by following coded instructions that come from the nucleus. Each ribosome, in its own way, is like a small machine in a factory, turning out proteins on orders that come from its boss, the cell nucleus. Cells that are active in protein synthesis are often packed with ribosomes. So, what does a ribosome do? Well, ribosomes takes DNA from the nucleus, which gets, and then turns it into ribosome. The ribosome turns the DNA into protein. All right. Endoplasmic reticulum. Eukaryotic cells also contain an internal membrane system known as the endoplasmic reticulum. Internal means inside, membrane means small barriers. The endoplasmic reticulum is the site where lipid components of the cell membrane are assembled, along with proteins and other materials that are exported from the cell. The portion of the ER involved in the synthesis of proteins is called the rough endoplasmic reticulum, rough ER. What does the rough ER do? Great quiz question. What does the ER do? Another good quiz question. What would it be like in real life? Be like the post office. The ER sends things in and out of the cell. Yeah. Okay. Newly made proteins leave these ribosomes and are inserted into the rough ER, where they may be chemically modified. Let's continue reading. Read with me to page 178. All right. Now we're going to learn about a new organelle. Notice we're learning about different organelles, different machines in the factory, different players in the team of the cell. The Golgi apparatus modifies, sorts, and packages proteins. Notice the stack-like membrane that make up the Golgi apparatus in this transmission electron micrograph. All right, so it's kind of a stacked pancake thing. Structurally similar to the ER, but what's different? There are no ribosomes stuck to it. Let's read what it does. Okay. Proteins are released or exported from the cell, synthesized on the rough ER, as are many membrane proteins. Rough ER is abundant in the cells that produce large amounts of protein for export. Other cellular proteins are made of free ribosomes, which are not attached to membranes. Other portions of the ER is known as the smooth endoplasmic reticulum. Because ribosomes are not found on its surface, in many cells, the smooth ER contains collections of enzymes that perform specialized tasks, including the synthesis of membrane lipids and the detoxification of drugs. Remember when we learned about lipids? What were those? Those were fats. Liver cells, which may play a key role in detoxifying drugs, often contain large amounts of smooth ER. The Golgi apparatus, now we're talking about that. Proteins are produced in the rough ER, move next into an organelle called the Golgi apparatus. You see, okay, did you catch that? Information comes from the nucleus. Ribosomes on the rough ER make the proteins. Then the proteins are moved to the Golgi apparatus. Discovered by the Italian scientist Camillo Golgi, as you can see in figure 7-9, Golgi appears as a stack of closely opposed membranes. Opposed means facing each other. The function of the Golgi apparatus is to modify, sort, and package proteins and other materials from the endoplasmic reticulum for storage in the cell or secretion to the outside of the cell. The Golgi apparatus is somewhat like the customization shop, where the finishing touches are put on proteins before they are ready to leave the factory. From the Golgi apparatus, proteins are then shipped to their final destination throughout the cell or outside of the cell, and this is really amazing. There's little address molecules that say, hey, this little protein has to go over to this part of the cell, or over to that part of the cell, or even be shipped out. Way cool. Golgi apparatus, the finishing shop. Rough ER, kind of like the assembly line. Nucleus, what was the nucleus again? Move on with me to page 179. Are you taking notes? Are you pausing the video from time to time to write down important information? I hope so. Let's read about lysosomes. Even the neatest, cleanest factory needs a cleanup crew, and that's what lysosomes are for. Lysosomes are small organelles filled with enzymes. Remember, enzyme is a protein that makes a chemical reaction happen? One function of lysosomes is the digestion or breakdown of lipids, carbohydrates, and proteins into small molecules that can be used by the rest of the cell. Lysosomes are also involved in breaking down organelles that have outlived their usefulness. Lysosomes perform a vital function in removing junk that might otherwise accumulate and clutter up the cell. A number of serious human diseases, including Tay-Sachs disease, can be traced to lysosomes that fail to perform their function. Ooh, what is Tay-Sachs disease? You want to write me a half-page paper on that and tell the class what it is? Give you some extra credit points. How much? It'll make it worth your while. All right? Let's read the next part. Vacuole. All right. Vacuole. Every factory needs a place to store things, and the cells cells contain places for storage as well. Some kinds of cell contain sac-like structures called vacuoles. What is that stores materials such as water, salts, proteins, and carbohydrates. What were carbohydrates again? Remember, they were sugars, long chains of sugars. Oh, wow, what a great question. What do vacuoles do? What do lysosomes do? What do Golgi apparatuses do? Great quiz questions. This is mainly a vocab chapter. In many plant cells, there's a single large central vacuole filled with fluid. The pressure of the central vacuole in these cells makes it possible for plants to support heavy structures, such as leaves and flowers. Vacuoles are also found in some single-celled organisms and in some animals. The paramecium in figure 7-10 contains a vacuole called the contractile vessel. Okay, here's the paramecium. Here is the vacuole. It's called the contractile vesicle. Okay. By contracting rhythmically, this specialized vacuole pumps excess water out of the cell. The control of water content within the cell is just one example of an important process known as homeostasis. Homeostasis, ding, 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 this is a great vocab word, and a quiz question, is the maintenance of a controlled internal environment. Great quiz question. Mitochondria and chloroplasts. All living things require a source of energy. Factories are hooked up to the local power company, but what about cells? Most cells get energy in two ways, either from food, molecules, or from the sun. Mitochondria. Nearly all eukaryotic cells, including plants, contain mitochondria. Mitochondria are organelles that convert the chemical energy stored in food into compounds that are more convenient for the cell to use. Mitochondria are enclosed by two membranes, an outer membrane and an inner membrane. The inner membrane is folded up inside the organelle. One of the most interesting